My name is Jamie Williamson. I'm Executive Vice President here at Scripps, and I want to be the first to welcome you to our Front Row Lecture Series. I always like to ask, how many people is this the first time that you've been to Scripps? Okay, lots of new people, and then everybody else has seen the show before, so you're really in for a great treat. I'd like to say just a couple things for those of you who may not be so familiar with Scripps, but we're one of the largest biomedical research institutes in the world. And whoa, there's somebody. And it's 2 campuses. We have a campus here in La Jolla. There's about 150 faculty. And we have another campus in Jupiter, Florida, with another 50 faculty. We have 5 departments that span both of the campuses. We have chemistry, neuroscience, structural biology, immunology, and molecular medicine. So we have a terrific graduate program. So we have 200 PhD students in all these disciplines of chemistry and biology. And we're very proud that that program is ranked in the top 10 consistently by US News and World Report. There are 2 sub-institutes I'd like to mention within Scripps. One is the Scripps Research Translational Institute, and that's led by Dr. Eric Topol. And they focus on genomic medicine, digital medicine, and launching a new initiative in artificial intelligence for biomedical sciences. The other institute was founded by our president, Pete Schultz, and that's the Caliber Institute. And it's a not-for-profit drug discovery institute where we try to accelerate production of medicines into the greater good for health. Now, I want to mention one other thing, that we were really thrilled last week to host Alan Alda here. And Alan was, obviously, well, anybody over 30 knows who he is. But in the second career, he has become an advocate for scientific communication. And he's really made a major effort in trying to get scientists to communicate better to the general public. And that's, in fact, one of the main purposes of the Front Row Lecture Series. But we're really happy that we were able to establish a partnership with his Institute for Scientific Communication. And he's going to be offering training here in California. They have another campus at Stony Brook University on Long Island, and we are going to be the West Coast presence for the Alda Center. And that we had a wonderful evening last week, and we're really happy about this new partnership. That's all I'm going to say by way of introduction, except to introduce our speaker today, and that's Lisa Stowers. She's a distinguished member of our neuroscience department. Now, everyone knows there's this central connection between smell and behavior. I mean, you smell a chocolate chip cookie and you go to the kitchen. I mean, there's this fundamental, this sensation that you have induces some kind of behavior. In fact, the entire industry centered around perfume and cologne is predicated on the fact that you can modify behavior through the sense of smell. So this is something fundamental that everybody in the room can understand. And Lisa's research is trying to understand the signals. When you smell something, how can that modify your behavior, your social behavior? And then also trying to understand those signaling pathways. And then how does the brain actually work and integrate this sensation and help you decide on what to do, mapping out pathways and understanding neurons in the brain. So I'm not going to say any more. I'll let her introduce herself and her brilliant lecture. So Lisa, please come to the stage.
So thank you for coming here today to Scripps. It's a great privilege for me to do science at Scripps. I love my job, and I'm very excited to be able to share with you some of the reasons why I think neuroscience is just a fabulous and interesting field, and a little bit about the type of work that we're trying to do to better understand the brain. The brain itself has really posed a mystery, and it's really one of the last organs or last regions of our body that we've been able to understand. When you look at other pieces of the body, such as the heart here, form indicates function a little bit. You can look at this, you can see it's a muscle, people observe that it beat, and you can sort of get an idea that it's used to circulate blood. But when you look at the brain, it's just this big glob of fat. It doesn't have a lot, there's not a lot that distinguishes it. It's mushy, it's got some shape to it, but the shape doesn't really indicate what it does. And so for the longest time, people had trouble really understanding what it was. And so if we look back into ancient Egypt, the Egyptians, they had a practice of preserving those that could, those that had the resources, preserving what they needed to be able to take to the afterlife. They believed that an afterlife existed, and that to be able to be successful there, you needed to bring stuff with you. And so if you had a pet, you would preserve your pet, some food, some money and jewelry, and then the body needed to be preserved because you needed a body with you. And they took great care to preserve all of the organs in little ampules, but the brain, they just pulled out and threw away. They thought it wasn't good for anything. So if there is an afterlife, those poor pharaohs are brainless right now, which is kind of a shame. If we jump forward to the time, even the 4th century, where Aristotle, these ancient Greeks were able to understand so much about the world, about biology and about physics, but he thought that the purpose of the brain was really just to cool the blood, that that was sort of an indication that if you were hot-tempered, that had something to do with the brain, but it really had no role other than just this passive sort of mechanistic one. And around that same time, Hippocrates was able to ascribe it as a seat of intelligence, and that kind of stuck. So since then, we've been able to study it for what it is. The other thing to think about is why we have a brain. And if you look at the other most common life form besides animals are plants, and no plants have a brain. And that really roots them in place, right? And so they're subject to the environment that they start in. They're just stuck with it. So whether there's good resources or bad resources, and whether there's danger or predators, they can't move away. And so I would really argue that one of the purposes of the brain is to help us move. And so because of a brain, we can go out and find food. We can hang out with people that we like. We can move away from predators. And that because of the brain, we're able to navigate very difficult and dense environments, some needing a lot of skill. And we do this with ease. Even little babies can do a lot of the stuff that's shown here, right? And so, so you may think, you need your muscles to do this, you need your heart to do this, and this is all true, but it's really because of the brain that we can do it. So to be able to move around, you have to be able to sense the world and figure out what's out there, figure out how you're going to move around it. You have to have a memory for whether the place that you were in was a place that was rewarding or whether it was a problematic. You need to be able to get up and move around. You need to have some sort of a driver, and that we sort of call our emotions. They're going to sort of drive you and reinforce the fact that you need to move. And you need to be able to sense what's going on in the rest of your body, whether you're sleepy, whether you're hungry, whether you have a hurt foot or a hurt back. All of these are going to affect movement. And really, so the point of the brain, I would argue, is that it allows you to decide, what should I do at any given moment, and then to be able to do it, that thing that plants can't do. The difficulty is that our brain is just a bag, as I said, of fat and water and salts and proteins and some chemicals. And so how does it figure out what's out there and how you should respond? And so what it's really doing is it's taking, from the sensory point of view, it's taking physical matter and transforming that into a biological representation, meaning it's taking everything that you can sense and turning it into a pattern of cellular activity. So just to look at this a little bit deeper, right? So we see things with light waves and we hear things with sound waves, but what we're getting in this seeing and this hearing that seems so real is really just a biological illusion. And so light waves, the small part that we can see, they form a continuum of waves. We can only detect and see a very small part of it. And we assign them a color from red to purple, but the color isn't inherent in the waves. It's not their purpose. They're a wave. And it's just your brain that has somehow creatively ascribed it a color. Most animals don't even see in color. There's just a very small subset like us that are fortunate and allow us this greater sense of how we can distinguish things in the world. But that's just an illusion, something that your brain has created. Same with sound. They're really just really tight waves or sort of long and slow waves. And the ability for us to hear something really high-pitched or very low-pitched is all just an illusion of our brain ascribing this different neural function. Moreover, the chemical senses are even crazier in that we can have a huge range of all sorts of different smells. Our tastes are more limited, but they're very different and distinct. And these are just molecules that are out there. The molecules themselves don't have an odor. They can float in the air, or they can bind other molecules, but they don't inherently have an odor. That's just a trick of our brain that allows us to distinguish them. So when you think of the rich smell of caramel or the distinctive taste of a raspberry, that's just your neurons in your brain that are tricking you and allowing you to have this sense so we can distinguish things. But the molecules themselves, or the matter in the world, doesn't really have that characteristic. And in fact, it's not even certain whether you and I smell raspberry the same. Your raspberry may smell like my dirt, and your dirt may smell like my onion or something, but we use them experientially, and we grow to like them or not like them based on the experience with them. And even moreover, our brain can do cool things. So if at the sense of taste, we only sense a few modalities: bitter, sweet, salty, sour, umami, which is the taste of protein. Some people think there's minor tastes like metallic, but they're very limited. And actually, all the flavor that we get from food that we feel on our tongue, and we know that we're eating it, it's part of what's happening right in our mouth. That's actually all coming from the nose, and your brain is taking what it's getting from the nose and what it's getting from the tongue and gluing them together so that you can have this wonderful sensation of flavor of food in the mouth. And in fact, if you get a cold, so that you are limiting this, the food tastes very different and may even completely lose most flavor depending on how strong your cold is. And that just tells you that they really are, you're actually smelling with your nose and tasting with your mouth, and your brain is gluing them together. Okay, so once you have a sensation, like here, if we look at touch with this coffee cup, how does your brain get that image? And so here you have that force. So here in this sense, it's a mechanical force, a pressure or a temperature. And if we zoom way into the tip of this finger, the cells, and at the very end of the cells, there's the cells themselves, which are tiny, maybe the size of the tip of a pin. They have a skin on them too, and we'll just call that the cell membrane here. And they have an outside and an inside. And then they have these proteins that sit inside them, and the proteins act like doors. And when they're closed, the cell is just like any other cell in your body. It just sits there like a skin cell or like a muscle cell, and it's not especially special. But once the door opens, and in this case, it's when force gets pushed on it that sort of pushes the door open. In the case of if you're smelling something, it's like a lock and key, where the odorants or the smells are like keys, and these proteins form a lock, and if they fit together, then they open. And that sort of works with light waves and hearing as well in that way. And once they open, ions rush in. And these are little things that we think of like salt. And they rush in, and when they do, it turns the cell, an electrical impulse will move down the cell. And that's what makes a neuron special, is that it can have this electrical activity. And from that, it can say that it sensed something way at the end, and if it's sensing this cup of coffee here, it could be the end of a finger. And in this case, it's a very long cell that goes all the way up to the spinal cord. And once it creates that electrical activity, it can move very fast, so almost as fast as you touch the coffee cup. The signal will move like an electrical current up all the way through your arm until it gets to the next cell. And at that point, the electrical signal will be coming in, and it'll change that into a chemical signal to be able to jump to the next cell. The electricity doesn't jump. And those chemicals can then either turn on the next cell. They're like the lock and key that causes the electrical signal to move down this cell. Or it can turn it off if it was already turned on. And this cell could say, hey, it's time to be quiet. So it can either turn it on and turn it off. And what I've told you so far, the cell biology of a neuron, we understand as neuroscientists and as a medical community, we know a lot about how this works. It's sort of how all the other cells in your body works, whether we want to understand how muscles are working or even how a tumor is working. We understand at least that much, if not more, about how neurons work to sense things and how their signals propagate and how they talk to each other. And so two neurons together is something we really understand. But what makes the brain especially difficult to study is that it's not just two neurons together. And what makes it special is that it's a bunch of neurons together. So each of these sort of wiggly, different colored things is a different neuron, just an artist representation. None of this is real. And they each have one end, which is sending signals, and another end, which is receiving signals. And they could be connected just in a simple way here, like to one cell, but they can also be receiving signals and sending signals to up to thousands of cells in the brain. And it's that ability of them to quickly send and receive signals at the speed of electricity across a certain pattern of who they're talking to, up to just a few neurons or a bunch of neurons, that makes it very difficult for us to study. Because when we look at them all, some of them have different shapes, and some of them have slightly different molecules, but they all look relatively the same. And they all behave sort of relatively the same. You can turn them on and turn them off, and what they do is they just send signals. And so how they're working to be able to allow us to see things or hear things or have good behavior is just a complete mystery. Moreover, it's not just this amount of neurons. They're in this structure called the brain. There's 86 billion of them about in your brain, and many of them are active right now, even though you're just sitting and listening. And we think it's really the patterns of activity that they are generating, which neuron is talking to when, and how much it's talking, whether it's talking a lot or a little, that makes the difference between smelling or tasting caramel or driving your car or just passively sitting here. And so it's really this problem here that makes neuroscience hard and hard for us to understand. More recently, we're in the world of computers. We've developed artificial intelligence, which is modeled a little bit on the brain, but really the pieces of the brain that's modeled on are not necessarily this complex in this 3D structure, acting in ways that we don't understand. So even though we can use the brain to help inspire artificial intelligence, we don't understand the brain enough to actually create an artificial brain or real artificial intelligence. Okay, so in my lab, we want to study the brain in 3D and all the things that it can do. And as a model, what we use is social behavior. And we like social behavior because it really is using, we think, almost all the different types of computations or different types of the way that the brain works, sort of all at once. And so it really take in sensation. So you guys entered the room and you saw seats and you knew you didn't have a ticket for a specific seat number and you knew that you should sit down sort of anywhere. And so that you used all of the cues that you had been given to know what to do, and everybody did it just great. So we all use sensation. And then social behavior is really dynamic. So it's an interplay between two individuals. So you have one sending signals, you receive it, and then you send some signals back, and you don't really know what the other person's going to do, and it sort of plays out in real time. That's what they do. It's going to inform what you do, what is going to inform, again, what they do again. And so it's very dynamic, and it's really using the full power of the brain in a really, really nice way. So we like that. It requires the brain to know how you're feeling, because that'll alter how you respond and you perform social behavior. So if you are stressed, if you are hungry, if you just learned something super duper awesome, all these things are going to affect how you respond to others around you. And so you really need to pay it, your body's paying attention to that. And then it also has to have a really good memory. You knew what to do in this room because you've been in rooms like it before. And so your brain sifted through everything and sort of found a pattern that matched it and said, this is what I'm supposed to do. And so you're doing that while you're doing social behavior as well. So social behavior in humans is hard to study. And one of the reasons is because it's all based on our experience, and we don't have a map of all our past experience, so we can't predict what you're going to do next, right? And that sort of requires past experience, means that it's culturally dependent too. So we have a cultural sort of norm that when we get into this environment in an elevator, we behave a certain way. It's not this way. And it would feel sort of weird, right? This is like a crazy day where this is happening for him. It's not normal, right? And so the other reason why social behavior is hard to study in humans is that it's really hard to map what's going on in the brain or even record what's going on in the brain during social behavior. So our best way to look at humans' brain while the brain is active is either in an MRI or a PET scan. Some of you may have had this before. And this is a way of looking at either the way that small molecules are moving, or the way the blood is moving around the brain, and it records an image of that. And when neurons are working extra hard, sending those electric activity very rapidly, they tend to use more blood, and so that is sort of a measure of neural activity. And so here you can look at somebody sitting in one of these MRI scanners, and you can look and you can see that we get these great patterns that change with what the person was asked to do. So this person was asked to listen, and here they're remembering, and they're very, very different patterns that we get, and we've learned a lot about how the brain works and how the human brain works by doing this. But it only works when a person is very still. So and the machine is very big and clumsy, and you can't really lug that around with you. And so doing really complicated tasks or any sort of natural behavior, besides having memories or a visual task or an auditory task, is very hard in this sort of situation. It's also not very sensitive. And so this is composed of quite a few points of light. And each one of them is this three-dimensional representation called a voxel, and that has about almost over a million neurons in it. And so each little dot of light here is representing a lot of different kinds of neural activity that's just sort of being smudged over. And then finally in a human, as you guys know, it's not ethically okay for us to go into your brain and move things around and ask what's going to happen. And as a scientist that really wants to get down to figuring out exactly why things are working, it's very helpful if we can go in and look and see what's happening, and then go in and change things and say, does this make it different? Does this make it better? Does this make it worse? Until we finally understand it. And once we can manipulate it, and we get out an answer that we had expected, that means we understand what's going on. And so that's really the gold standard, and we can't do any of that in a human. Okay, so we use animals, and social behavior is a great thing to study in animals because they do it. And you can look at these images and sort of get a sense of perhaps what they're doing, why they're doing it, based on the fact that we do similar things too, as well. And so most of these sort of very simple social behaviors are highly conserved across evolution. I've seen some really cool videos of starfish fighting each other. They do it really slowly, but they are very aggressive. So even things that you don't think move around very much and have good social behavior, once people go looking for it, they usually find a little something there. Okay, the other thing that we like about studying animal social behavior is that it's largely innate. So unlike trying to learn about what's going on in this room or figuring out what to do in an elevator where that's culturally dependent, most of the behavior in animals, they all do it the same way. And so if they're all doing it the same way, without variation, sort of to the same cues and with the same sort of scaling and response, that's a really good thing for science, because we can sort of look across individuals if we put them in the same environment, we're going to get the same thing. Unlike humans, where we would get a different thing with every variations of the same thing with different individuals. And we believe that if you're seeing the same thing, that means that your brain is acting in a very similar way. So even though you may have all come in and sat down, what drove you to sit down? Maybe you were feeling tired, maybe your foot hurts, or maybe you were just really excited to hear me talk, you know. So those are all different reasons, and you're gonna have different brain activity patterns with those different reasons. But if these guys are doing it because they have the same genes, and their neural activity is the same, then we can study that. From one individual to the other individual, it's going to be reproducible. And the reason that we think that it's hardwired, that they're doing it the same way, that it's innate, and that they're going to have the same activity, is because we think that there's certain neurons in the brain that are pre-programmed to do this, and that they're expressing certain genes. And that we can go in and find those genes and pull them out, and we can understand why they're doing this sort of thing. Now, humans don't do it exactly the same way, and so our genes are there to sort of set a level of things that we're born knowing how to do. Babies know how to get milk from their mother. They're just born knowing how to do that, and that would be one human behavior that is entirely innate. But the rest of our behavior, there's sort of some things to it, but the rest of it you need sort of some prodding and pushing and practice and trial. And so we think that the genes that we find in animals are going to be similar to what's in humans, and then humans have an extra special layer on top of it that we can figure out later. Okay, some social behavior in humans, though, we do think is innate, and these are things that we think when they go extremely awry, and we can see they deviate from our normal behavior. So I'm not one to believe that there's exactly one type of normal behavior. I think behavior is a full continuum, and that everybody's a little bit somewhere here and there on it. But at the very tails of the continuum, sometimes behavior becomes so different from everybody else that it can cause trouble and make it more difficult to be able to integrate and to perform easily. And so some of these examples of these would be different features in the autism spectrum, and we do know that there are certain genes that are prone to drive these sorts of differences, and there's many labs in the world that are using those genes and modeling them in animals and to being able to understand why, so that ultimately we can rationally figure out how to make those problems go away. HGHD is also another one where we think that there's certain genes that predispose a person to having a more difficult time focusing. And then a very extreme example would be something like schizophrenia, also where we think genes underlie these sorts of behaviors. These are the types of things that are very extreme. A lot of these types of social behaviors that they're affecting are specific to humans, either with language or with the way that we sense and socialize. And so those sorts of things are more difficult to model in animals. And so my lab has chosen not to use those particular cases to model, even though these are the types of questions that ultimately we would like to find an answer to understand how the brain is responding differently. And instead, we are interested in studying sort of more normal social behavior, understand that first, and then figure out the differences when things are fully expressed. Okay, so I've been talking a lot about neurons, and the other thing that's in the brain that's very important to social behavior is actually hormones. So here's, this is a very, very general sort of way to think about hormones. So some of these are hormones, other of these we call neurotransmitters, which are those chemicals that travel in between the neurons. And in general, if you read the popular literature, you probably know that we refer to oxytocin as the love hormone. And it definitely has an effect, especially in animals, between parents and offspring, or between mating partners, and it facilitates them. But it also does other things. It was originally found for its role in generating the ability of moms to make milk, to be able to feed their young. And other aspects of pregnancy and parition. And it does it has other general functions in the brain. And that's common between all of these neurotransmitters and hormones, is that they have a role in behavior. the way that we understand them is they don't do just one thing, they do many things. And sometimes, depending on the context or the way that we look at them, a single thing, even like serotonin, can do entirely different things in the brain. And then all of these things also have roles outside of the brain in just controlling your heart rate and whether your pupils will dilate or your digestion or your ability to sleep. And all of these things come back and feed into behavior, and we'll talk about that a little bit later. But it just shows you that these have a very wide variety of roles, and we don't fully understand. Most of the time we're studying them one at a time, and we don't understand how they're all working in concert at any given instant to be able to regulate neural function and generate behavior. And so the last point that I want to make, the sort of general point, is that because of all of these unknowns, we really don't know how the brain is working in 3D. We really don't know which patterns are important at every given moment. And we don't know how these really important molecules are feeding in to turn on and to turn off neurons and change the patterns, and what sort of a normal level should be, and what happens when they get off. And for those reasons, we don't really know how to treat diseases or differences in the way that the brain functions. So currently in the United States, about 1 in 3 of us have taken some sort of medication for generally to modify our moods, generally for stress or anxiety or depression. That's a lot of people. That's a lot of things. And so we kind of think that if we have a problem, we can just go to the doctor and they'll fix it. But as you may know, it's not so easy even to fix it, even when we sort of understand the underlying cause. Each prescription has to be personalized, and they work better for some than others. And part of that is because we really don't know how stress, anxiety, or depression is generated in the brain. We can see those patterns in the MRI scanner, but we can't get down to the neurons and understand exactly what's changed in those neurons to be able to say, we should go in and change the levels of serotonin or dopamine or a neurotransmitter like GABA. And so because of that, what we're doing with these drugs right now is, instead of pouring oil into the right place, the right specific place in the car, our drugs are like oil, and we're just pouring them all over the car. We're pouring them all over the brain. And the brain, we think, has certain places in it that are going to alter your mood and regulate whether you're feeling anxious, and other places in it that also have the ability to detect these molecules, this oil, but they're doing something entirely different and used in different circumstances. And if you pour the oil everywhere, it's not always going to work to give you the thing that you like without some side effects. And so what we want to do is do more basic research to understand how the brain is working in 3D, to understand what are the rules that make it, that enable it to be able to generate the right behavior at the right time that's appropriate for you. And then once we understand those rules, then we can go in. And I think with the chemists and all the other biology that we know, we should be able to figure out a way to pour the oil in the right place and pour the right type of oil in the right place without sloshing it around and ultimately get there. So again, it's very difficult. We don't know very much. This sort of thing is not the type of thing that's going to happen in the near future. If anything, it's a goal to try and get to in the far future. Okay, so my lab wants to study this. We want to understand how the brain is doing this. Instead of using humans, we use mice. They're they have a brain that's very much like a human, but they're 3 orders of magnitude smaller than a human, and they have awesome social behavior. And the types of things that we want to know that social behavior does, we think as neuroscientists are sort of coming through the brain in sort of a predictable order. And so first there's the sensory information, and the individual wants to know what's out there and how much of it there is. And so say there is that smell of chocolate chip cookie that Jamie was describing earlier. It first wants to know, is there a cookie? And then it wants to know, is there a cookie, and is there a sign that says cookies $10. And if you see that, you may not want the cookie anymore, right? So, it just depends on whether, what the sensory information is. You want to ask yourself, have you ever had a cookie? And did you like it or did you not like it? And that's going to inform what you're going to do next. You want to ask yourself if you're hungry and whether that sounds like a good idea. Or if you're hungry but you're about to go on a run and you really don't want to have that in your stomach, thinking about what your internal state needs are at the moment. And then after all of this, so first sensation, then the brain's going to send the information through a lot of different places in the brain and decide what to do about it. And we imagine that there's some center in the brain that's, although it could just be a network property, but we imagine that there's a place in the brain that's then going to tell the muscles and the hormones what to do. Should I go to the counter and order a cookie and then start to eat it? If so, maybe I should start my stomach enzymes going. Maybe I should start to get a little saliva in my mouth. So the brain's going to prepare for both the motor action and what your body needs to do to get ready for it. And we imagine that then that goes out into the brain and the new sensations coming in, and that this loop is just iterated all the time. And so, so I told you that several times now that the environment is going to drive, and experience is going to drive how we respond. And I keep sort of alluding to that there's this other thing in you, this internal state that's also driving you how to respond. And just as shorthand, we're going to call that emotion. And that's the thing that's going to drive you. And this, like the brain, is sort of this construct that we don't really understand what it is or how it works. We just know that it feels really strong, it can be very demanding, and that it's very important. And so as recently as Shakespeare's time, they really didn't understand these emotions at all. They imagined how they worked. They imagined that people could be divided into sort of 4 different types. These were based on earth, wind, rain, and fire. And they assigned different sort of general personality types, which were driven by emotions. And the brain was only responsible for one of these, and in fact, it's probably the most boring type of personality, if that doesn't, I don't mean that insultingly, but it's sort of the most plain one. And then other aspects of emotional behavior were attributed to the spleen or the heart, these sorts of things. So we all now know that's very silly. And that we really think it's the body that is driving, that the brain is driving the body, and then the body is responding and telling the brain what should be happening. And so, but this is sort of complicated. And again, this is very similar to what I said about the magic that happens when the brain is able to ascribe a particular sensation, that sort of magical sensation, that taste of caramel, gets a feeling. How we get these feelings, we don't really know. So some of the indicators that we get are something like you could have a tight stomach and you could not be able to focus, right? But there's many times where you get those same symptoms and they mean entirely different things. And so it could mean that you're sick. It could mean that you're really in love. It could mean that you're nervous about something, or even that you're hungry, right? And your brain's using all these other cues and its past experience to be able to say what that tight stomach means. But it's that interplay of your brain interpreting what your body's doing and then trying to override it, the brain trying to then tell the body what to do, and then interpreting the meaning. And we really think it's sort of this heart-racing, sweat-producing breath rate that is telling your brain what emotional state you're in. But the brain has driven that emotional state, and we don't know how or why. Okay, so that's sort of the conundrum of emotions. I think I explained it very confusingly, but it's sort of a chicken and an egg problem. So we can't study any of this in a mouse because we can ask him if it's hungry by just giving him food, but we can't ask him if he feels hungry. But these sorts of things are very difficult. This is where you really want to do this sort of work in people. But in the 1980s, psychologists, sort of led by Russell, imagined that almost all of human emotional drives could be explained by just two axes that sort of generate four different quadrants. And the first is how much you like something, whether you don't like it or whether you like it, so whether it's good or bad. And the second axis is what your response is to it. Is it just a blah response, or are you really aroused and excited by it? So whether it's mild to intense arousal. And so if you sort of are able to focus on any of these different things, you can see that if you're astonished, you just won the lottery, you know, that could be very joyful, and it's highly intense, and it's up in this quadrant. You know, and so these sorts of things, again, we can't ask a mouse whether it feels gloomy or is amused, but we can ask it whether it likes something or doesn't like something. And then we can measure its pupil dilation, or whether it's moving around a lot. And we can ask whether it is not aroused or whether it's aroused. And so these characteristics are sort of what we use in science to be able to, and if we can measure them, it's slightly aroused and is slightly unpleasant, then we can imagine that we're modeling something similar to this, although we don't imagine that the mouse is feeling something similar to that. Okay, so my lab is asking how the brain is doing this. We want to look at how much it likes something or doesn't like something. And these are the types of things that we eventually want to be able to figure out where in the brain is happening. And we imagine that once we understand where things are happening, then we can start to look at the molecules and the genes and figure out how they are happening. And so the first little bit of science that I'm going to tell you about is the story of attraction. And as we all know, the females, you can imagine, female mice like males, and they're attracted to each other. And in the lab, they're not always attracted to each other, and that's based on their ovulation cycle. And so in the lab mice, it's really quick. Every 5 days, they go through an entire cycle. So on day 1, they're ovulating, and at that point, they're attracted, and we call that estrus. And then just 2.5 days later, they are in the exact opposite phase of the cycle. They are not interested in males at all. And we can see that by their behavior, and they respond just to the smell of the male. So if we put the smell of the male on one side of the cage and a control that doesn't have a smell on the other side of the cage, and then we ask where they move around and see whether they like it or they don't like it, when they're attracted to a male, they like the smell of the male. And all these blue lines are where they've walked, and where it's warmer, these reds and yellows are where they sat and where they walked more. So they spent more of the time over here. And just a couple days later, the same female, when she's given the smell, she doesn't like it so much. She spends less of the time, more time over here, okay? And throughout the years, we've been able to purify these molecules, and we know that the male smell has these gray blobs that are very attractive. And so during the estrus cycle, she's paying attention to these, and during the diestrus cycle, part of the cycle, she's not. And so we wanted to ask how the brain was changing as it was smelling the male, and what was the pattern of change? And that would tell us whether she liked something or didn't like something, we were hoping. And so, but before we started looking into the brain, the postdoc that was working with me said we should really look at the nose and make sure that the nose isn't also changing throughout these 5 days. And I said, well, that's ridiculous. The nose won't change, because the purpose of a sensory system is to just gather as much data as possible. And whether you're attracted to a male or not attracted to a male, you still want all the data. It should come in, right? And the function of the brain is decision making. And that's when we should decide what we should do with that information. And we know this to be true. And you've experienced this a lot. And the easiest example is called the cocktail party phenomenon. And in that, you have a crowded room, and people are making so much noise that you really, it's hard to hear. But you can focus on someone in front of you, and you can attend to them, and you can have a nice conversation. And everything else sort of recedes into a din. But you can, if somebody across the room says your name, you may hear it. You may know what direction it's coming from, whether a male or female said it, and if you know the people very well, you may even know who said it. And so that means when everything was receding to a din, everything was getting in, but your brain was being a filter. It was filtering out the stuff it didn't need and just focusing on this guy, right? And that's how we expect everything to be in all the sensory systems. But this experiment was easy to do. And so what she did, we can look at the nose outside of the brain, and we can take those cells and just put them under a microscope. And we can also, the two things that change primarily between when she's attracted and when she is not attracted is that during the attraction phase, she has a lot of estrogen in her body. And when she's not attracted, she has a lot of another hormone called progesterone, here in gray. And if we take away the ovaries, she doesn't have either. So we can look at the response after we add back one of the hormones, and if we add them both back and the response doesn't change, then we're good. But in fact, the response changed. So each of these lines represents the electrical activity. We're seeing it in a different way, but it's really the neuron that's being active, as I told you before, after it sensed that attraction smell from the male. And so here's a bunch of neurons, and they sense it, lock and key, they sense it, lock and key. But then we add back the hormone that's around, the progesterone that's around when she's not attractive. We wait a short period of time, and she doesn't respond. These neurons don't respond at all. They just shut off. And we thought, well, maybe they're dying in the dish, because that could happen. They're outside the brain. But we do a little trick where we turn them on, and they all turn on. We turn them on in a different way, and they all turn on just fine. And so that tells us that this progesterone changed these neurons. Okay, so these are nose neurons. And what we have before, so again, this is like the skin of the cell that I showed you when we were talking about how you sense touch. And instead of having that door protein, now we have another lock and key. It's this molecule here, and it's when it senses the smell from the outside, it tells the inside of the cell through this relay mechanism of these proteins. This is the gate that allows those ions to rush in and the electrical impulse to go. And that's what's happening when we add this attraction pheromone. Go, go. But we also found that there's this other receptor that's here, lock and key, and it's sensing progesterone. And it's doing it in such a way that when progesterone is around, it sends a message to this molecule that turns it off. And when it turns it off, that's what's happening here. And so it doesn't even fire. Because this guy's off, this guy never gets turned on, and the electrical activity never, ever comes. Okay? So what we found is really that normally, when there's no progesterone around, the female can sense everything. And it gets a representation, including these attraction guys, and that there's a sense of them, the brain knows they're out there, and it starts to become attracted, and it moves the body towards the smell. Okay? But just a couple days later, when she has that progesterone around, it's not as though the brain gets the activity and decides it doesn't want to use it. Instead, out there in the nose, what's happening is that the neurons themselves are not active. The progesterone shuts them off. So the brain never even gets the signal. She doesn't even know that the male smell is out there to be able to respond. So she can never act. She can never do the behavior. This is an all or none gating of the behavior, and it makes social behavior in these sorts of species very robust. But when I learned this, I thought this was ridiculous. Because as I told you before, the nose isn't there to make decisions. That doesn't happen. The brain is there to make decisions, and I want my brain to get even more information. I want to see things I can't see. I want more information, not less. And so I was told by a lot of other scientists, oh, this makes a lot of sense, because if the females were sensing this male odor when she wasn't in a state where she was ready to reproduce, then she would just be distracted by it. And I thought that was ridiculous, right? Because we're getting all sorts of sensory information all the time, and we can, as with the cocktail party phenomenon, our brain is so good at tuning things in and tuning things out. We hear of sensory overload, but that's not so much of a thing. We can get tired and we bounce right back if we get a little more sugar in us and we're good, right? So I thought that was silly. And so what we found is we've gone along and studied it, and what that was just the tip of an iceberg. And it turns out this whole system in mice has a bunch of receptors there, the locks, and for a bunch of molecules that act as the keys that are able to turn on and off the sense of smell in the mouse. So they have a bunch of things that respond to these keys that are the stress keys, and these guys that are responsive whether you're hungry, that tell your body whether you're hungry or whether you're not hungry. And so it's a whole system that's in place in the mouse. We're not sure if this is there in people. We see some of these receptors there, but we're going to figure it out a lot more in the mouse before we go off and look at people. So right now we imagine that we are all not subject to this sort of system. But the other thing that it told us, and the reason why I'm telling you about this, even though it's not in the brain, is that our idea that the periphery, the edges of the brain were doing simple things, like sensation or talking to muscles. And in the middle of the brain was doing the hard stuff, the decision-making, the thinking about it. And what we find from this story is that the decision-making doesn't even have to be in the brain. It can be outside the brain. And so that opens up the ability for us to think without these boundaries and to be able to imagine that any of these things could be happening anywhere. And we're looking for these patterns that could be happening anywhere. Okay, so I have talked too much, and I have one more story to tell you, and I'm going to tell it to you really fast and see if I can do it in just the next minute or so. Let's see if I can go fast. And so the other thing that we're doing is we want to be able to study everything that's responding to social behavior as an entirety. And we still think that's only a very small portion of the brain. And that right now, we can't study the whole brain at once. That's too much. And so we look at one region of the brain, and we think that's like blind man touching an elephant. You know, somebody sees a tree or a wall or a snake, and that, you know, you see a center for love and a center for stress, and we imagine that there could be other things going on, and if you're only looking here, you don't see the bigger picture. And so, so we have found a behavior that's a very simple behavior that we think is going to give us the bigger picture. And so this is a male now. Before I was telling you about females. When a male smells a female here, what he does is he starts to sing to her. And he sings to her in a language that we can't hear. It's ultrasonic. It's really high. So if you were around, he would just be quiet because our ears can't detect it. But other females can hear, and they like it. And so this is the behavior that we look at. And we like it because it's super simple. It's not like some of those other complex behaviors that lots of motor groups happen. It just uses the larynx and the lungs. What I wanted to show you is that we found a very small number of neurons, just these red neurons in the brain. There's just a few thousand of them. And we can turn them on by using this light. And so you can see the light goes on. And this is a drawing of those noises that he's making. This is a sonogram. These are like the sound waves. And so when we turn it on, he starts to make those noises. And those are just these, we turn it on with that light, and the light goes all the way into the brain and turns them on right there. Special trick. And we can turn them off too. And we really like studying this behavior because it's very simple. And we think that there are features that are very much like the types of vocal communications that we use, the babies use when they're crying, from a little cry to a strong cry, or that we use when we laugh, a little polite laugh versus a belly gut laugh, and that mice are doing this too. And I wanted to show you this cat video. So what I want to remind you of is that our pets, yeah, you can imagine. So this little guy is howling, but he's going to be very clean afterwards, and everybody will be happy. And so our pets scale this information, and the mice do too. And so, and we figured out how that worked. And so these are the new neurons those red neurons that we turned on and made calls. And we knew a little bit something, other people have known something about how we produce vocal noises just based on human language and studying human language. And so our new node is A, and we plugged it into B all the way down. And we wanted to ask, well, why is there an A? What makes this special? And what we found, and I'm going to speed this up and actually not show you the data, is that there is this new region of the brain allows them to it allows them to be flexible in the calls, just like that cat or your pet can go from a little noise to a big noise. And that without it, the brain just makes very strong, hard noises. It doesn't have that ability to be able to scale, and it's that scaling that we think is important in social communication and in emotional response so that we tell each other how we're feeling on the inside. And what we found was that it's not magic. It's really just the way that the neurons are organized and the way that the neurons respond after they're turned on and off, which you can't really understand because I'm not telling you the whole story. But there's this variable response. They're not all doing the same thing. And by them not doing the same thing, it leads this variability to the simple behavior of just calling. And so, so what we think is that by using the social behavior in the mouse, we can understand new ways of the way the behavior is organized. We can study individual nodes and ask what they're good for and what makes them different. And then ultimately, we're hoping to find things that hormones and other things interact with, and we can understand how they change when the animal is doing appropriate social behavior and when they're not doing appropriate social behavior. And when they're not doing the appropriate social behavior, how we can design therapeutics to be able to better bring them up to the normal social behavior. And so this was all done with a team of really fabulous graduate students and postdocs. These are all young people that are aspiring to do neuroscience, and they come to Scripps to be able to learn how to do it. We've worked with a range of collaborators, both here at Scripps and around the world. And we've been supported by funds here at Scripps, as well as from the National Institutes of Health. Thank you very much. Sorry.
Okay, thanks very much. Now, before we go out to reception, we have the opportunity for people in the audience to ask Lisa some questions. And I would really encourage you to take the advantage of the opportunity to ask. There's no such thing as a dumb question, but if you manage to come up with one, I'll point it out. But no, no please. So I have a little bit of a hard time seeing it. Please, question.
Do you think that there's been cognitive revolution over the past 40 years, particularly in the last 40,000 years, particularly in the last 10,000 years, when we have evolved very rapidly in our social evolution, cultural evolution?
Yeah, I mean, that's a good question and one that's really difficult to answer. We don't have a fossil record or a record of what brains were like at that point in the past, right? We can look and see, just how much change the environment puts on behavior. My daughter's now on this little square phone device for many hours every day, and this is something that we never did before. We used to be very physically active, and now we spend a lot of time in the Western world being very sedentary. And these could affect all sorts of different things. Whether the brain already had all the magic to do all that, and we're just using it differently, or whether there could be a pressure to evolve and make it different. I think that's something that's I don't have the answer to, and that may be difficult to figure out.
What are the implications for your studies for the concept of free will?
It's an excellent question. If I take sort of the most reductionist interpretation, my interpretation of what free will isn't, it would be that our brains are sort of like little machines, and the information comes in, and our genes and proteins are going to act on it a certain way, and an outcome will come out. We may think that we have control over that outcome, but the genes and proteins made it happen just one way, no matter what. And that would be my sort of definition of no free will. And the other free will would be the information comes in, and then I get to actively decide which way the information is going to go through my brain and where I want it to land in order to do something. Is that sort of an okay interpretation of free will? Okay, so based on that, my imagining, from what I know about the brain, is that genes and proteins, sort of what sets up what a cell can do, what shape it's going to be, who it talks to, how it responds, what sorts of locks it has on it so that it can respond to different things, and what sort of keys it sends out. Those sorts of things are determined by genes and proteins, and that they are set. But human behavior, we also have all of this experience. And that experience is a very powerful influence on our genes and proteins, and it can change them. And so if you're under stress for a long period of time, it will change the types of genes and proteins that are expressed in your neurons, and that makes them respond differently and send out different messages. And so, so with all that being said, whether we have the ability to reroute the information, you know, I kind of think that when I'm making an intellectual response, you know, I'm solving a problem, I can see a lot of different answers and I choose the correct one, and I'm not sure whether the genes and proteins had that predetermined. So I'm going to walk a fine line here and say, I think that a lot of things that we do may not necessarily believe what we want to call free will, that our genes and proteins are doing them, that's based on our past experiences, but that we probably do have the ability to sort of reroute our attention and what the brain's doing, either you know, moving us to a different location and getting a new environment or something that may be like free will. It's going to be hard to figure that out.
Is there any consistency in the functional MRIs of humans as opposed to animals? Is there any use for statistical information?
Statistical.
Across human, whether there are patterns that are repetitive across
Right.
human, not
Right.
in a specific.
So in those functional imaging, a lot of it has been reproduced from across humans. So a lot of our common understanding that there are different areas of the brain that do different things, and that there are different patterns that are evoked, whether we have different feelings inside of us. Those are all fairly reproducible across humans. And then when we go into the animal models, and so even in mice, and we look for, so if we find a region that's activated during a stressful thought in a human, we can go back and we can find those regions of the brain in mice that are especially dense in receptors that are expressing stress hormones. And so there is the ability to translate, whether we can use statistics between the two or statistics across the patterns and make a case for what's real and the amount of conservation. I don't think we can do that.
I was just wondering when you said that that actually the brain transferring colors and smells is really an illusion. Now, I work with seniors and the elderly, and most of the time these people are transitioning between dementia or in stages of Alzheimer's. And always, the first complaint is they complain that they can't taste things or they don't enjoy the way things taste. And I'm wondering, is that a function of brain, the diminution of the brain activity or something else?
Yeah, we do know a little bit about that. So in the neurodegenerative disorders, the types of changes that take place in the brain, the olfactory region is especially sensitive to it. So there's regions that affect more recent memories. Those are also affected. Regions that affect your emotional state, those are affected. And the region that affects your smell is one of the earliest regions that sort of decays. And there are efforts to be able to use that as sort of a sentinel to be able to identify the onset of neurodegeneration. And there are ideas right now, people trying to generate stem cell replacements and things like that to be able to replenish, especially the olfactory system, because that can drive feeding and energy levels, and sort of have a whole range of repercussions. If food doesn't taste good, things, the quality of life can go downhill. And so there are efforts to maintain that. So we don't think that the neurodegenerative diseases are targeting these places, but there's something about them where these areas are especially sensitive.
Excuse me?
Right in the light.
Hi, thank you. I was wondering if you believe that we only use a small percentage of our brains, and if things like supplements, mental exercises, or anything else actually has a significant impact. Thank you.
Yeah. So I think when I was growing up, I heard that we use only 10% of our brain at any given time, or that, or maybe, I think it's sort of at any given time, right? It's not that we only ever use 10% of our brain. So that's a question, you know, the imaging studies that we see, those images that you see where you only see certain regions lined up, and sometimes you can see if a person's really resting, a lot of it is very cool colors, just that blue. They never light up. But what that is actually is there's sort of controls that are done. So the person gets into a scanner and there is some activity, and those are subtracted out, and then the experiment starts. And if you add those back in, you sort of wash out everything. There's sort of activity everywhere. So that's sort of our first clue that it's a lot's happening. If you put electrodes on your brain, there's a lot that you hear. It's very noisy. Lots going on. And now, just recently, with the Brain Initiative, one of the goals is to be able to look at all the neurons and their activity in the human brain at one time. And that goal is still pretty far off. But in some very small animals, we can now do this. And so if we look at small fish that are transparent and we can see through their brain, we can look at neural activity the whole time. And at that point, we see patterns that are like fireworks going off all the time. And so we don't know. In our social behavior, we've been able to measure activity, and we find some areas that are very, very quiet until the animal is in a social situation. And so we don't really know. We don't know enough about how the brain works so that I would recommend using a supplement, particularly for the brain, or not. If there's no bad side effect, it's not going to hurt you, and we don't know enough to say that it's not good for you or it's not doing anything. But like the drugs, it would be nice eventually to be able to find something that has a targeted action and isn't just acting everywhere. You had a third point, and supplements. Exercise, right. So exercise is just good for everything. You're getting more blood that's moving around, and so that's gonna, I often do things, I'm right-handed, and I'll often do things like unload the dishwasher or brush my teeth with my left hand, just because I think it's good for my brain to get something new. I think your brain always getting something new is a good thing. You're challenging it, but whether it actually prevents old age memory loss, I'm not sure that we know that. It's just a good idea to always challenge your brain.
Okay, we'll do one more question right here.
Hello. Can you hear me? Okay. Yes, my question has to do with an indirect measurement of neural activity in the brain. I understand, I'm not a neuroscientist, but I understand that the gut has the second largest aggregation of neurons in our body. And so could you, by monitoring the microbiome of a person, and this wouldn't be instantaneous correlation, but could you determine what's going on in a person over a period of time, such as Alzheimer's disease or something like that, by monitoring their microbiome?
So there's an active area of research that's looking into how the gut is talking to the brain and how the brain is talking to the gut. And there definitely is a connection. Right now, I don't know the literature well enough to say whether there's a good general consensus. It's hard to imagine a case where we could use the gut microbiome, or that's the bacteria that's in your gut, whether we could use that to say exactly what the brain's doing. But we can definitely look at the nervous system in the body and use that as an indication of what the brain is doing under some circumstances. And that's also an area of active research, even here at Scripps.
Okay, well, I think, I'm very lucky that I work at Scripps and I get to come to work every day and learn something. I'm glad you could come here and share that and get some insights into what's going on here, and to get a window into Lisa's brain. So let's thank her for this brilliant lecture.